Hello, this is Gina Piscatelli with the second lecture on the autonomic nervous system for students in Anatomy and Physiology 1 at Madison Area Technical College. In this lecture, we'll talk about the anatomy of the autonomic nervous system and its neurotransmitters. So the first anatomical characteristic that I want to talk about relates to the pathways of both sympathetic and parasympathetic that begin in the central nervous system and then travel out to the periphery. So we're going to start with motor pathways in the autonomic nervous system. Each subdivision begins in a very distinct part of the central nervous system. And you might recall that somatic motor neurons are located in the brain, the spinal cord, the brainstem, and the spinal cord. So all regions of the central nervous system. That's somatic motor, voluntary motor. But autonomic motor only begins in very distinct or subsections of the central nervous system. So that's one thing. The other thing that's different between somatic and autonomic is that the pathways for autonomic contain two motor neurons. The first one starts in the central nervous system. And so that neuron leaves the central nervous system and it synapses on the second neuron. So there's what we call a preganglionic neuron and a postganglionic neuron that will synapse in a structure that we call the ganglia. And we'll look at an anatomical view of that in a second. So there's two motor neurons. There's only one motor neuron that controls skeletal muscle coming from the central nervous system. So those somatic motor neurons are quite long. We've got two in the pathway for the autonomic nervous system. So the parasympathetic nervous system begins only in the brainstem or in the sacrum. So it's shown here on your left. The first neuron will originate either in the brainstem or in the sacral spinal cord. Of course, in both locations, you've got multiple neurons. Therefore, it's often called the craniosacral division. The sympathetic nervous system, in contrast, the first neuron originates within either the thoracic or lumbar regions of the spinal cord. So notice that there aren't any autonomic neurons beginning in the cervical spinal cord. Not at all. And I also mentioned that one difference between somatic and autonomic is that there are two neurons in an autonomic pathway. So we'll take this first example on the top here. I have a parasympathetic pathway listed first up on top. And this is going to innervate some visceral structure in the head or the neck, maybe a salivary gland, okay, or a blood vessel, and the smooth muscle in a blood vessel. So the first neuron originates in the brainstem, that's the cell body, and the axon goes out to the effector, the organ. But before it actually enters the organ, there's another neuron that takes over the signal. So the first neuron synapses on the second neuron. And this is the synapse, and that's called a ganglion. So it's surrounded usually by connective tissue, and there's usually more than one synapse occurring in that location. The second example I have is sympathetic. Sympathetic, as you know, would originate either thoracic or lumbar cord. I'm showing like low thoracic here. The first neuron extends out with its axon, and notice that the first neuron is really short and the ganglion is very close to the spinal cord. Very, very close. So the second neuron is much longer, and it innervates whatever organ. I've got it on the intestine in this example. And just to be complete, I listed another example of a parasympathetic pathway that begins not in the brainstem but in the sacral spinal cord. The same thing is going to be true. The first neuron is going to be very long, almost reaching, if not completely reaching, the organ that is to be innervated. But just before innervation happens, there's another neuron that takes over the signal. That's the postganglionic neuron, number 2. And this time the ganglion is very, very close to the organ that's being innervated, as was true up in the head region. But it is not true for sympathetic pathways. Sympathetic pathways, the ganglion is very close to the spinal cord. So if we look at a parasympathetic pathway, this is a different picture, but we see that it originates either in the brainstem or the sacral cord. The first neuron, the preganglionic neuron, is very long. The second neuron, the postganglionic neuron, is very short. And sometimes there's a little ganglion that's close to the organ of innervation, like there's a ganglion close to the eye, close to the salivary glands. But sometimes the ganglion is actually in the wall of the organ, like within heart tissue, within the wall of the stomach, within the wall of the bladder. And so the second neuron is actually traveling within the wall. So if this is the wall of the bladder, it's got a certain thickness. The second neuron would actually begin within the wall. It's embedded in there and travel throughout the wall, sending out signals. So we often call those ganglia intramural ganglia. Now the sympathetic pathway is a little bit different. The beginning of a sympathetic pathway will start either in the thoracic or the lumbar spinal cord. And the first neuron, called the preganglionic neuron, will travel a short distance. In this diagram, it's dark green. The first neuron is dark green. And then the second neuron, the postganglionic neuron, is longer and it's light green. Okay, so it's longer. Therefore, the synapse between the first and the second neuron happens in some ganglia that are very close to the spinal cord, and in fact, they're part of a chain of ganglia. We call it the sympathetic chain ganglia. And then, of course, the postganglionic neuron goes to innervate whatever organ we're talking about, be it something in the head, something in the thoracic or abdominal cavity, or pelvic. There are some examples of other ganglia associated with the sympathetic pathway that are not part of this chain. They're called the collateral ganglia, but they're still, generally speaking, not so close to the organ of innervation. They're still closer to the central nervous system. Okay, this is just a summary of the two different pathways and what we've talked about so far regarding the differences in anatomy between the parasympathetic and the sympathetic division. So in terms of where they begin, these pathways, for parasympathetic, the beginning is either in the brainstem via a cranial nerve or the sacral spinal cord traveling within a sacral nerve. For sympathetic, the beginning, the first neuron, is traveling within a spinal nerve that started in either the thoracic or the lumbar spinal cord. So that's one thing. The next thing that we talked about was the neurons and how long or short they are. So there are always two neurons, and in the parasympathetic nervous system, the first neuron is long, the second neuron is short. But in the sympathetic nervous system, it's the opposite. The first neuron, the preganglionic neuron, is short, and the second is long. So the second neuron is much longer. Therefore, that sets up a different location for the ganglia. So the ganglia are also located in different, depending on which division you're talking about. So in the case of the parasympathetic nervous system, ganglia are always close to the organ of innervation. Many times we would call this ganglion an intramural ganglion. Other times it's just called a parasympathetic ganglion. In the sympathetic nervous system, the ganglia are close to the spinal cord, or the central nervous system, I should say. There are some cases where there are sympathetic ganglia that are not part of that chain. They're a little bit off. They're called collateral ganglia. They still are never in the wall of the organ, though. So that's a review of what we've talked about so far. Now we're going to look at function. So there are three factors that account for why the functions are opposite in the sympathetic and the parasympathetic division. One factor is that often an organ has different parts, and each part is innervated by a separate division. So different cells in the same organ are innervated. The example I'll show you has to do with the size of the pupil. Another factor is that each division uses a different neurotransmitter. So if you recall, neurotransmitters can either be excitatory or inhibitory depending upon what ion channel they open. So if an ion channel that allows sodium through, I'll say sodium here, if that's what the neurotransmitter opens, then that's excitatory. So here's my neurotransmitter. It opens. That's excitatory because you're going to get positive charge on the inside. If instead what happens is the neurotransmitter opens a chloride ion channel, this is just review, that's a negatively charged ion, and the neurotransmitter binds. So it could be, it's a different neurotransmitter. I should make it a different one. Make it a different color. Different neurotransmitter binds. Okay. Now what you'll get is negative flow in, so you'll get inhibition. So different neurotransmitters can be either excitatory or inhibitory. Now to complicate things even further, it is possible that the same neurotransmitter can open both of these, but a cell will only have one of them. So this neurotransmitter could be purple, and this neurotransmitter is also purple. So what are you going to say? Is the neurotransmitter excitatory or inhibitory? Well, it's both, but it depends on where you are in the body, which channel is there. That's the easiest way to think of it, or which receptor is there. So I'll show you an example of that. So first, different parts of an organ are innervated by the sympathetic versus the parasympathetic. And the example I want to use is the iris. So the iris of the eye is the colored part of your eye, and it contains actually two muscles, smooth muscles. One is radial and one is circular. So if I look at this picture, it's easiest to see that you have some muscle fibers coming out radially, but you also have some muscle fibers that are circular. So the sympathetic nervous system, as we know, causes the pupil to dilate. The reason why is because it innervates the radial muscle. And so the radial muscle contracts. That means the fibers shorten. Contraction always means shortening in muscles. And so the pupil dilates. The parasympathetic, by contrast, we know, constricts the pupil. And so the muscle that the parasympathetic innervates is the circular muscle. And so it acts like a sphincter. The muscle's rounded, but when it contracts, it kind of comes together or squishes together, and that makes the pupil constrict. So that's one reason that the two branches can have opposite effects, is different parts of the iris are controlled by each division. Another reason that the two divisions can have opposing effects has to do with what neurotransmitters they release. For example, we know that the parasympathetic nervous system makes the heart rate go down. So we need it to inhibit cardiac muscle contraction. What does that is the neurotransmitter acetylcholine, and that is what the parasympathetic nervous system releases to the heart. The sympathetic nervous system, in contrast, uses the neurotransmitter epinephrine, and that excites the cardiac muscle. So you get an increase in the rate of contraction in the heart. Now, one thing I want to point out has to do with the other neurotransmitters. So the one we're talking about is the postganglionic neuron. That's the one that affects the target organ. So, oops. Boy, it just doesn't like that. Okay, so the postganglionic neuron is what's releasing the acetylcholine or the epinephrine, right? But there's a preganglionic neuron that I guess we should consider. And in that case, acetylcholine is released by both. And acetylcholine is excitatory to the postganglionic neuron, just to be complete. And one thing that's really important is we know that acetylcholine is inhibitory to cardiac muscle. But do you remember that acetylcholine is excitatory to skeletal muscle? Always. So the parasympathetic nervous system doesn't innervate skeletal muscle, right? It only deals with cardiac and smooth muscle, so you don't get conflicting problems. Okay, the last reason that the two divisions can do different things, or that one division, like the sympathetic nervous system, can have what seems like opposing effects in different organs. And that has to do with the fact that really the action of a neurotransmitter depends on the receptor that binds to it. And there are lots of different receptor types for epinephrine in particular. And epinephrine, of course, is sympathetic. So we are talking about sympathetic. Okay. So another term for epinephrine would be adrenaline. And there is a difference between epinephrine and norepinephrine, but we treat them as the same. We don't get into the pharmacology of the difference. So epinephrine is fine, or adrenaline is fine. There are some alpha-adrenergic receptors and there are some beta-adrenergic receptors. Some of these receptors, when they bind to epinephrine, cause excitation. In other cases, the receptor, when it binds to a neurotransmitter, epinephrine, inhibition occurs. So you have to think about the fact that you have a cell, the target cell, that has a receptor on it for epinephrine. And so epinephrine would bind to the receptor and cause some kind of an effect. What it causes, what the receptor's activation causes, is what varies. Will it be an excitatory stimulus or is it an inhibitory stimulus? And that depends on the receptor itself. And I mentioned one way you can think about that is like what ion would go in. That's one way to think about it. We also talked a little bit about G protein linked receptors. So those have varying effects. So let's look at the circulatory system, in particular blood vessels, and how the sympathetic nervous system can cause vasoconstriction in some blood vessels, but vasodilation in other vessels. Because that's the big thing that the sympathetic nervous system can do for us, is it can divert blood away from the digestive tract to our skeletal muscles and our heart. Okay, so in skin and digestive organs, there are alpha-1 adrenergic receptors. So when epinephrine binds to these receptors, the cell is excited. So if this is in the skin and, it's in the blood vessels of skin and digestive organs, and they have been excited because epinephrine bound to their receptors. When they're excited, they constrict. So you have less blood flow to the skin and digestive organs. Now I'm going to take the other example, beta-2 adrenergic receptors. When these receptors bind to epinephrine, there's inhibition in the target cell. And we see these receptors in blood vessels that supply our skeletal muscles and our heart. So this adrenergic receptor is in the heart. When epinephrine binds to it, wait a second, that doesn't make any sense. In blood, better take the skeletal muscles. In blood vessels that supply skeletal muscle, if epinephrine binds to that receptor, then you would have inhibition of the muscle and you would get vasodilation. Once you have vasodilation, more blood flow is going to go to the skeletal muscles and the heart. So let's look at a sort of a visual example versus all those words. Okay, so we're always talking about blood vessels. That's the important part. It's always blood vessels that have this, what seems like, contrasting effect by the sympathetic nervous system. The sympathetic nervous system sometimes is excitatory, sometimes inhibitory, sometimes causes vasoconstriction, sometimes causes vasodilation. So in the skin, blood vessels that have these alpha adrenergic receptors. When epinephrine binds to those receptors, you get excitation and contraction, so less blood flow. If we think about the blood vessels that exist supplying blood to the skeletal muscles, they have different receptors. Those are called beta adrenergic receptors. Now, when epinephrine from the sympathetic nervous system finds these blood vessels and the epinephrine binds to the receptor, now you have inhibition. They're inhibitory. The muscle does not contract, and instead the vessel dilates. So more blood flow to the skeletal muscles. I know that's a little confusing. You've probably heard about beta blockers, for example. Okay, so if I go back to this slide and I'm talking about blood vessels that supply blood to skeletal muscle and to the heart, right? The beta blockers typically are acting on these adrenergic receptors. And the objective in some cases, not all cases, because beta blockers can be against beta 1, beta 3, beta 2, whatever. The heart blood vessels are in danger of rupturing, so you don't want too much blood flow going through there. So if you block the receptor, they don't dilate quite as much. And we're talking about blood vessels that are on the surface of the heart that actually supply. I'm trying to draw with this thing. It just does not like me at all. Okay, that's okay. But nonetheless, we get more blood flow going to the heart and the skeletal muscles. And the blood vessels are on the surface of the heart. Okay, that's the end of autonomic nervous system. Thank you very much.